Good day everyone. This video will talk about bioenergetics, enzymes, and metabolism. In bioenergetics, we will focus on endergonic and exergonic reactions. A living cell bustles with activity, such as macromolecules of all types are assembled from raw materials, waste products are produced and excreted, genetic instructions flow from the nucleus to the cytoplasm, ions are pumped across cell membranes, and so forth. To maintain such a high level of activity, a cell must acquire and expand energy. The study of the various types of energy transformations that occur in living organisms is referred to as bioenergetics. So there are two concepts that is linked in bioenergetics, which is energy and thermodynamics. So energy is defined as the capacity to do work, while thermodynamics is a study of the changes in energy that accompany events in the universe. Now let us discuss the first law of thermodynamics. The first law of thermodynamics is the law of conservation of energy. It states that energy can neither be created nor destroyed. Energy can, however, be converted or transduced from one form to another. Cells are also capable of energy transduction, storage, and transport. The chemical energy stored in certain biological molecules such as the ATP or adenosine triphosphate is converted to mechanical energy when muscles contract, to electrical energy when ions flow across a membrane, or to thermal energy when we shiver in the cold. The most important energy transduction in the biological world is the conversion of sunlight into chemical energy, the process of photosynthesis, which provides the fuel that directly or indirectly powers the activity of nearly all forms of life. To discuss energy transformations evolving matter, we need to divide the universe into two parts: the system under study, or a certain space in the universe, or a certain amount of matter, and the surroundings, or the remainder of the universe. Even though the system may lose or gain energy, the first law of thermodynamics indicates that the loss or gain must be balanced by a corresponding gain or loss in the surroundings, so that the amount in the universe as a whole remains constant. The energy of the system is termed the internal energy, and its change during a transformation is delta E. One way to describe the first law of thermodynamics is that the change in internal energy is equal to the difference between the heat energy and its mechanical work energy. Depending on the process, the internal energy of the system at the end can be greater than, equal to, or less than its internal energy at the start, depending on its relationship to its surroundings. In other words, change in internal energy can be positive, zero, or negative. Let us consider a system to be the contents of a reaction vessel. As long as there is no change in pressure or volume of the contents, there is no work being done by the system on its surroundings, or vice versa. In that case, the internal energy at the end of the transformation will be greater than that at the beginning if heat is absorbed and less if it is released. Reactions that lose heat are termed exothermic, and ones that gain heat are endothermic, and there are many reactions of both types. Because the change in internal energy for a particular reaction can be positive or negative, it gives us no information as to the likelihood that a given event will occur. To determine the likelihood of a particular transformation, we need to consider some additional concepts. The second law of thermodynamics expresses the concept that events in the universe have direction. They tend to proceed downhill from a state of higher energy to a state of lower energy. Thus, in any energy transformation, there is a decreasing availability of energy for doing additional work. For example, rocks fall off cliffs to the ground below, and once at the bottom, their ability to do additional work is reduced, and it is very unlikely that they will lift themselves back to the top of the cliff. Similarly, opposite charges normally move together, not apart, and heat flows from a warmer to a cooler body, not the reverse. Such events are said to be spontaneous or naturally occurring, a term that indicates that they are thermodynamically favorable and can occur without the input of external energy. The loss of available energy during a process is a result of tendency for the randomness or disorder of the universe to increase every time there is a transfer of energy. This gain in disorder is measured by the term entropy, and the loss of available energy is equal to T delta S, where T is temperature and delta S is the change in entropy between the initial and final states. Entropy is associated with the random movements of particles of matter, which, because they are random, cannot be made to accomplish a directed work process. According to the second law of thermodynamics, every event is accompanied by an increase in the entropy of the universe. The release of heat, for example, from the oxidation of glucose within a cell or from the friction generated as blood flows through a vessel, is another example of an increase in entropy. The release of thermal energy by living organisms increases the rate of random movements of atoms and molecules. It cannot be redirected to accomplish additional work, because the energy of molecular and atomic movements increases within or with the temperature, so too does the entropy. It is only at absolute zero or 0 Kelvin when all movement cease that the entropy is zero. Together, the first and second laws of thermodynamics indicate that the energy of the universe is constant, but the entropy continues to increase towards a maximum. The concepts inherent in the first two laws were combined with American chemist J. Willard Gibbs in 1878 into the expression, where delta G is the change in free energy, that is, the change during a process in the energy available to do work. Delta H is the change in enthalpy or total energy content of the system. T is the absolute temperature, and delta S is the change in the entropy or entropy of the system. The equation states that the total energy change is equal to the sum of the changes in useful energy and energy that is unavailable to do further work. When rearranged and written as, the equation provides a measure of the spontaneity of a particular process. It allows one to predict the direction in which a process will proceed and the extent to which the process will occur. All spontaneous energy transformations must have a negative change in free energy. That is, the process must be proceed toward a state of lower free energy. The magnitude of change in free energy indicates the maximum amount of energy that can be passed on for use in another process, but tells us nothing about how rapidly the process will occur. Processes that can occur spontaneously, that is, processes that are thermodynamically favored or have a negative change in free energy, are described as exergonic. In contrast, if the change in energy or change in free energy for a given process is positive, then it cannot occur spontaneously. Such processes are thermodynamically unfavorable and are described as endergonic. The signs of change in enthalpy and the change in entropy for a given transformation can be positive or negative, depending on the relation between the system and its surroundings. So, if the change in enthalpy will be positive if heat is gained by the system, and negative if heat is lost. The change in entropy will be positive if the system becomes more disordered, and negative if it becomes more ordered. Now that we have discussed the concepts of free energy in general terms, we can apply the information to chemical reactions within the cell. All chemical reactions within the cell are reversible, and therefore we must consider two reactions occurring simultaneously, one forward and the other in reverse. According to the law of mass action, the rate of a reaction is proportional to the concentration of the reactants. Consider, for example, the following reactions. The rate of the forward reaction is directly proportional to the product of the molar concentrations of A and B. The rate of the forward reaction can be expressed as K1AB, where K1 is a rate constant for the forward reaction. The rate of the backward reaction equals to K2CD. All chemical reactions proceed, however slowly, toward a state of equilibrium, that is, toward a point at which the rates of the forward and backward reactions are equal. At equilibrium, the same number of A and B molecules are converted into C and D molecules per unit time, as are formed from them. At equilibrium, therefore, in other words, at equilibrium, there is a predictable ratio of the concentration of products to the concentration of reactants. This ratio, which is equal to K1 over K2, is termed the equilibrium constant, or the KEQ. The equilibrium constant allows one to predict the direction, forward or reverse, in which the reaction is favored under a given set of conditions. Suppose, for example, that we are studying a reaction and we have just mixed the four components A, B, C, D so that each is present at an initial concentration of 0.5 m. The direction in which this reaction will proceed depends on the equilibrium constant. If the equilibrium constant is greater than 1, the reaction will proceed at a greater rate toward the formation of the products C and D than in the reverse direction. So for example, if the equilibrium constant happens to be 9, then the concentration of the reactants and products at equilibrium in this particular reaction mixture will be 0.25 and 0.75 respectively. If on the other hand, the equilibrium constant is less than 1, the reverse reaction will proceed at a greater rate than the forward reaction, so that the concentration of A and B will rise at the expense of C and D. It follows from these points that the net direction in which the reaction is proceeding at any moment depends on the relative concentrations of all participating molecules and can be predicted from the equilibrium constant. So if you will proceed forward or backward by writing out the equation for the equilibrium constant, then plugging in the actual concentrations of all the reactants and products, and then comparing this value to the expected value of the equilibrium constant, and if the result is equal to the equilibrium constant, then the reaction is already at equilibrium. If, however, the result is less than the equilibrium constant, then this means that the reaction will proceed toward the product formation. One special type of equilibrium constant is the dissociation constant, denoted Kd. The dissociation constant is a measure of how much two molecules tend to stick together. Given two molecules A and B, for example, a protein A that binds a ligand B, so the reaction is where AB is the complex of A bound to B. Usually this refers to a non-covalent interaction. For this reaction, the equilibrium constant is called the dissociation constant and is defined as because of change in free energy for a given reaction depends on the reaction mixture present at a given time. It is not a useful term to compare the energetics of various reactions. To place reactions on a comparable basis and to allow various types of calculations to be made, a convention has been adopted to consider the free energy change that occurs during a reaction under a set of standard conditions. For biochemical reactions, the conditions are arbitrarily set at 25 degrees Celsius or 298 Kelvin and 1 atmospheric pressure, with all the reactants and products present at 1 mole concentration except for water. The standard free energy change describes the free energy released when reactants are converted to products under these standard conditions. It must be kept in mind that standard conditions do not prevail in the cell, and therefore one must be cautious in the use of values for standard free energy differences in calculations of cellular energetics. The relationship between the equilibrium constant and the standard free energy change is given by the equation, and when the natural log is converted to log 10, the equation becomes where R is the gas constant and T is the absolute temperature. Recall that the log of 1 is 0. Consequently, it follows from the above equation that reactions having equilibrium constant greater than 1 have negative standard free energy change values, which indicates that they can occur spontaneously under standard conditions. In other words, given the reaction written as follows, if the standard energy change is negative, the reaction will go to the right when the reactants and products are all present at 1 molar concentration. The greater the negative value, the farther to the right the reaction will proceed before equilibrium is reached. Under the same conditions, if the standard free energy change is positive, the reaction will proceed to the left, that is, the reverse reaction is favored. One of the most important chemical reactions in the cell is the hydrolysis of ATP. In the reaction, ATP plus H2O yields to ADP plus P. The standard free energy difference between the products and reactants is negative 7.3 kcal per mole. Based on this information, it is evident that the hydrolysis of ATP is highly favorable or exergonic reaction, that is, only that tends toward a large ADP over ATP ratio at equilibrium. So there are several reasons why this reaction is so favorable, and One of it is that the electrostatic repulsion created by four closely spaced negative charges on ATP is partially relieved by formation of ADP. It is important that the difference between the change in free energy and the standard free energy change be kept clearly in mind. The standard free energy change is a fixed value for a given reaction and indicates the direction in which that reaction would proceed were the system at the standard conditions. Because standard conditions do not prevail within a cell, the standard free energy change values cannot be used to predict the direction in which a particular reaction is proceeding at a given moment within a particular cellular compartment. To do this, one must know the change in free energy, which is determined by the concentrations of the reactants and products that are present at the time. So in the equation, A, B, C, and D are the actual concentrations at the time. Calculation of the change in free energy reveals the direction in which the reaction in the cell is proceeding and how close the particular reaction in question is to equilibrium. So for example, a typical cellular concentrations of the reactants and products in the reaction for ATP hydrolysis might be ATP is equals to 10 millimoles, ADP is equals to 1 millimoles, and P is equals to 10 millimoles. Substituting these values into the equation, thus the change in free energy is equals to negative 11.5 kcal per mole. Even though the standard free energy change for the hydrolysis of ATP is negative 7.3 kcal per mole, the typical energy change or change in free energy in the cell for this reaction is about negative 12 kcal per mole because a cell maintains a high ATP-ADP ratio. Reactions with large positive standard free energy change values are typically driven by the input of energy. Consider the formation of the amino acid glutamine from glutamic acid by the enzyme glutamine synthetase. So, this endergonic reaction takes place in the cell because glutamic acid is actually converted to glutamine in two sequential reactions, both of which are exergonic. So, in the overall reaction, it has the standard free energy change of negative 3.9 kcal per mole. The formation of glutamine is said to be coupled to the hydrolysis of ATP, as long as the change in free energy for the ATP hydrolysis is more negative than the change of free energy for glutamine synthesis from glutamic acid is positive. The downhill ATP hydrolysis reaction can be used to drive the uphill synthesis of glutamine. To couple the two chemical reactions, the product of the first reaction becomes the reactant for the second. The bridge between the two reactions, glutamyl phosphate in this case, is called the common intermediate. What happens in essence is that the exergonic hydrolysis of ATP is taking place in two steps. In the first step, glutamic acid acts as an acceptor of the phosphate group, which is displayed by the ammonia in the second group. The hydrolysis of ATP can be used in the cell to drive reactions leading to the formation of molecules such as glutamine because ATP levels are maintained at much higher levels than they would be at equilibrium. This can be demonstrated by the following calculation. Thus, at equilibrium, the ADP concentration would be expected to be more than 10 raised to the 7th times that of ATP. But in fact, the ATP concentration in most cells are 10 to 100 times that of ADP. This is a crucial point because it's the relative concentrations of ATP and ADP that matter. If a cell were to contain an equilibrium mixture of the ATP and ADP, it wouldn't matter how much ATP was present. The cell would have no capacity to perform work. ATP hydrolysis is used to drive most endergonic processes within the cell, including chemical reactions such as the one just described, the separation of charge across a membrane or the concentration of a solute, and changes in the properties of proteins. ATP can be used for such diverse processes because its terminal phosphate group can be transferred to a variety of different types of molecules, including amino acids, sugars, lipids, and proteins. But in most coupled reactions, the phosphate group is transferred in an initial step from ATP to one of these acceptors and is subsequently removed in second step. Now let's talk about enzymes. So enzymes are the mediators of metabolism, responsible for virtually every reaction that occurs in a cell. Without enzymes, metabolic reactions would proceed so slowly as to be imperceptible. Even though enzymes are proteins, many of them are conjugated proteins. That is, they contain non-protein components called cofactors, which may be inorganic or metals or organic or coenzymes. When present, cofactors are important participants in the functioning of the enzyme, often carrying out activities for which amino acids are not suited. Enzymes exhibit the following properties. First, they are required only in small amounts. Second, they are not altered irreversibly during the course of the reaction, and therefore, each enzyme molecule can participate repeatedly in an individual reactions. And third, they have no effect on the thermodynamics of the reaction. Enzymes do not supply energy for a chemical reaction and therefore do not determine whether a reaction is thermodynamically favorable or unfavorable. Enzymes are remarkably adept catalysts. Catalysts employed by organic chemists in the lab, such as acid, metallic platinum, and magnesium, generally accelerates reaction 100 to 1,000 times over the non-catalyzed rate. In contrast, enzymes typically increase the velocity of a reaction from 10 raised to 8 to 10 raised to 13 fold or greater. In addition, unlike the inorganic catalyst used by chemists, enzymes are highly specific in the reactants they can bind and the reaction they can catalyze. The reactants bound by an enzyme are called substrates. How is it that an enzyme can cause a reaction to occur hundreds of times a second when the same reaction might occur at undetectable rate in the enzyme's absence? The answer lies in the formation of the enzyme-substrate complex, which allows the substrates to be taken out of solution and held into the surface of the large catalyst molecule. Once there, the physical and chemical properties of the substrate can be affected in a number of ways. So there are three mechanisms by which enzymes accelerate reactions. So first is maintaining precise substrate orientation. Substrates bound to the surface of an enzyme are brought very close together in precisely the correct orientation to undergo reaction. In contrast, when reactants are present in solution, they are free to undergo translational and rotational movements, and even those possessing sufficient energy do not necessarily undergo a collision that results in the formation of a transition state complex. Next mechanism is changing substrate reactivity by altering its electrostatic configuration. Enzymes are composed of amino acids having a variety of different types of side chains, from fully charged to highly nonpolar. When a substrate is bound to the surface of an enzyme, the distribution of electrons within the substrate molecule is influenced by the neighboring side chains of the enzymes. This influence increases the reactivity of the substrate and stabilizes the transition state complex formed during the reaction. These effects are accomplished without the input of external energy such as heat. The last mechanism is exerting physical stress on bonds in the substrate to be broken. Although the active site of an enzyme may be complementary to its substrates, various studies reveal a shift in the relative positions of certain of the atoms of the enzyme once the substrate has bound. In many cases, the conformation shifts so that the complementary fit between the enzyme and reactants is improved, or induced fit and the proper reactive groups of the enzymes move into place. This type of movements within an enzyme molecule provides a good example of a protein acting as a molecular machine. So, as these conformational changes occur, mechanical work is performed, allowing the enzyme to exert a physical force on certain bonds within a substrate molecule. This has the effect of destabilizing the substrate, causing it to adopt the transition state in which the strain is relieved. Enzyme inhibitors are molecules that are able to bind to an enzyme and decrease its activity. The cell depends on inhibitors to regulate the activity of many of its enzymes. Biochemists use inhibitors to study the properties of an enzyme, and many pharmaceutical companies produce enzyme inhibitors that acts as a drug. So, enzyme inhibitors can be divided into two types, the reversible and the irreversible. So, irreversible inhibitors are those that bind very tightly to an enzyme, often by forming a covalent bond to one of its amino acid residues. Reversible inhibitors, on the other hand, bind only loosely to an enzyme and thus are readily displaced. So, for the reversible inhibitors, we will discuss competitive and non-competitive types. Competitive inhibitors are reversible inhibitors that compete with the substrate for access to the active site of an enzyme. Because substrates have a complementary structure to the active site to which they bind, competitive inhibitors must resemble the substrate to compete for the same binding site, but differ in a way that prevents them from being transformed into product. In non-competitive inhibition, the substrate and inhibitor do not compete for the same binding site. Generally, the inhibitor acts as a site other than the enzyme's active site. The level of inhibition depends only on the concentration of the inhibitor. Increasing the concentration of the substrate cannot overcome it, because in the presence of a non-competitive inhibitor, a certain fraction of the enzyme molecules are necessarily inactive at a given instant, that the maximal velocity of the population of enzyme molecules cannot be reached.
Good day, everyone, and now let's tackle glycolysis and ATP production. First, let's identify what glycolysis is. Glycolysis comes from a merger of two Greek words. Glyco means sugar or glucose, lysis means breakdown or splitting. Thus, the literal translation of glycolysis is the breakdown or splitting of glucose. Glycolysis is the central pathway for the glucose catabolism, in which glucose, or the 6-carbon compound, is converted into pyruvate, or the end product of the glycolysis, or the 3-carbon compound, through a sequence of 10 steps. Later, we'll find out what are these steps. The primary function of glycolysis is to produce energy in the form of ATP, or the adenosine triphosphate. Similarly, glycolysis also produces pyruvate, which is then oxidized further to create more ATPs. Glycolysis occurs in the extramitochondrial fraction of the cell in the cytosol. This unique pathway occurs aerobically as well as anaerobically and doesn't involve molecular oxygen. Glycolysis can function with or without oxygen present. In glycolysis, it is either followed by fermentation or cellular respiration, depending on the availability of oxygen. The glycolytic sequence of reactions differ from species to species only in the mechanism of its regulation and in the subsequent metabolic fate of the pyruvate formed. In aerobic organisms, or in aerobic condition where oxygen is present, glycolysis is the prelude to citric acid cycle, or what is known as the Krebs cycle, and ETC, or the electron transport chain. The following are the enzymes that catalyze different steps throughout the process of glycolysis. Since glycolysis consists of about 10 steps, and each step has a different reaction that is catalyzed by a different enzyme. Glycolysis is the systematic breakdown of glucose to produce energy. Energy is released in the form of ATP and NADH. So we can see here is the 10 steps of glycolysis. The picture may look difficult or complicated, but later we'll have a stepwise explanation of these steps. But before that, I just want you to know that during glycolysis, a single mole of 6-carbon glucose is broken down into 2 moles of 3-carbon pyruvate by a sequence of 10 enzyme-catalyzed sequential reactions. These reactions are grouped under two phases, phase 1 and phase 2. For phase 1, or the preparatory phase, it is the first 5 steps of glycolysis. For phase 2, or as the payoff phase, it is the last 5 steps or reactions of glycolysis. For phase 1, energy is consumed, while for phase 2, energy is produced. Okay, now let's have the stepwise explanation of glycolysis. For step 1, or the phosphorylation of glucose, the first reaction that takes place is the conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. Hexo means 6, denoting the 6-carbon compound, and kinase means group of enzymes that catalyze the phosphorylation reaction. In this reaction, ATP is converted into a molecule of ADP, hence it utilizes one molecule of ATP. Step 2, we have the isomerization of glucose 6-phosphate. Glucose 6-phosphate is reversibly isomerized to fructose 6-phosphate by the enzyme phosphohexoisomerase. This reaction involves a shift of the carbonyl oxygen from C1 to C2, thus converting an aldose into a ketose. Step 3 is the phosphorylation of fructose 6-phosphate. This step is the second priming step of glycolysis, where fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the presence of the enzyme phosphofructokinase. Like in step 1, the phosphate is transferred from ATP while some amount of energy is lost in the form of heat as well. Step 4, we have the cleavage of fructose 1,6-diphosphate. This 6-carbon sugar is cleaved to produce 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by aldolase. Step 5, we have the isomerization of dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate can be readily degraded in the subsequent steps of glycolysis, but dihydroxyacetone phosphate cannot be. Thus, it is isomerized into glyceraldehyde 3-phosphate instead. In this step, dihydroxyacetone phosphate is isomerized into glyceraldehyde 3-phosphate in the presence of the enzyme triose phosphate isomerase. This reaction completes the first phase of glycolysis. Step 6, we have the oxidative phosphorylation of glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, NAD is reduced to coenzyme NADH by the H from glyceraldehyde 3-phosphate. Since 2 moles of glyceraldehyde 3-phosphate are formed from 1 mole of glucose, 2 NADH are generated in this step. Step 7, we have the transfer of phosphate from 1,3-diphosphoglycerate to ADP. This step is the ATP generating step of glycolysis. It involves the transfer of phosphate group from the 1,3-bisphosphoglycerate to ADP by the enzyme phosphoglycerate kinase, thus producing ATP and 3-phosphoglycerate. Since 2 moles of 1,3-bisphosphoglycerate are formed from 1 mole of glucose, 2 ATPs are generated in this step. Step 8, we have the isomerization of 3-phosphoglycerate. The 3-phosphoglycerate is converted into 2-phosphoglycerate due to the shift of phosphoryl group from C3 to C2 by the enzyme phosphoglycerate mutase. This is a reversible isomerization reaction. Step 9, we have the dehydration 2-phosphoglycerate. In this step, the 2-phosphoglycerate is dehydrated by the action of enolase, 2-phosphoenolpyruvate. This is also an irreversible reaction where 2 moles of water are lost. Step 10, which is the last step, we have the transfer of phosphate from phosphoenolpyruvate. Phosphoenolpyruvate is converted into an enol form of pyruvate by the enzyme pyruvate kinase. The enzyme catalyzes the transfer of a phosphoryl group from phosphoenolpyruvate to ADP, thus forming ATP. Okay, so we have here a summary of the process of glycolysis that can be written as follows. We have glucose plus adenosine diphosphate plus phosphate plus nicotinamide adenine dinucleotide to pyruvate plus water plus adenosine triphosphate plus nicotinamide adenine dinucleotide plus hydrogen ions. In this table, we can see that in step number 1 and 3, it is where energy is being consumed, while in step number 7 and 10, it is the energy generating step. There is a net utilization of only 2 ATPs since 4 ATPs are used in the second phase, but 2 ADPs are also produced in the first phase. There is a net use of only 2 ATPs, or there are 4 ATPs that are being produced and there are 2 ATPs that are being consumed. So therefore, we have a net gain of 2 ATPs. The overall process of glycolysis results in the following events. Glucose is oxidized into pyruvate. NAD is reduced to NADH. ADP is phosphorylated into ATP. And now, let's talk about ATP production. The fuel that provides energy in our body is called ATP, or the adenosine triphosphate. It is composed of adenine, ribose, and 3 phosphate groups. It transfers energy to many different chemical reactions. Almost all metabolic pathways directly or indirectly run on energy supplied by ATP. In human cells, cellular respiration releases energy from energy-rich organic molecules and changes ADP into ATP. Aerobic respiration is the main ATP-producing pathway. That's why, in order to make ATP, the cells in our body use a process called a cellular respiration. Anaerobic respiration produces much less ATP because no oxygen is involved. Aerobic respiration, which is the main ATP-producing pathway, first we have here is the initial breakdown of glucose through glycolysis that was discussed earlier. Next we have is the Krebs cycle, and the final stage of aerobic respiration occurs in the electron systems. ATP from anaerobic pathways. Anaerobic pathways operate when oxygen is absent or limited. Pyruvate from glycolysis is metabolized to produce molecules other than acetyl coenzyme A. When we say acetyl coenzyme A, it is the main function is to deliver the acetyl group to the citric acid cycle, or what is known as the Krebs cycle, to be oxidized for energy production. ATP production pathways. Glucose is one of the organic molecules that can be broken down to form ATP, or that can be used to produce ATP. So, how exactly is glucose used to produce ATP? The process has three steps. First, we have is the glycolysis. Glycolysis takes place in the cytoplasm where glucose is broken into three carbon molecules called pyruvate. Pyruvate, which is produced in the cytoplasm, enters the mitochondria and is converted to acetyl coenzyme A. Acetyl coenzyme A is a molecule that participates in many biochemical reactions in protein, carbohydrate, and lipid metabolism. Its main function is to deliver the acetyl group to the citric acid cycle to be oxidized for energy production. Next, we have is the citric acid cycle, or what is known as the Krebs cycle. It is also called a citric acid cycle because a citric acid is one of the important intermediate molecules. Krebs cycle of respiration requires oxygen. If oxygen is present, pyruvate is taken to the inner membrane of mitochondria and broken down. The Krebs cycle stores high energy electrons and produces 2 ATP. The final stage, we have the electron transport system or the electron transport chain. The production of ATP is completely dependent on the supply of oxygen that withdraws the electrons at the end of the transport systems. For the glucose breakdown, glycolysis yields 2 ATP, Krebs yields 2 ATP, and electron transport yields 32 ATP for a grand total of 36 ATP for glucose molecule.